know that bacteria are capable of producing specific polymers? Yes, indeed. With the help of enzymes, bacteria can produce high molecular weight biopolymers as polysaccharides, amides, polyphosphates, and polyesters from simple sugars, fatty acids, and amino acids. And why do they produce polymers? To better live and compete in the environment they are growing in. Very broadly, polymers may serve as an intracellular substrate reservoir, as a glue to attach to surfaces, and to form granules as a protective layer, or finally to increase water content and concentration of external substrates. Polymers can be present inside the cell, intracellular, or outside, extracellular. A variety of biopolymers are produced by bacteria during wastewater treatment. The most relevant and promising to be used so far are polyhydroxyalkanoates, PHA, and extracellular polymeric substances, EPS, that can be used in substitution of synthetic-based polymers. As for PHA, they are a class of biologically synthesized polyesters, including as much as 150 types of monomers that are randomly distributed in microstructures or organized as block polymers. The most common monomer being the 3-hydroxybutyrate, producing polyhydroxybutyrate, PHB. PHA are bio-based and biodegradable, thus classifying PHA as a promising group of eco-efficient bioplastics. Indeed, their market is displaying a high relative growth rate as reported by the European Plastic Association. PHA biosynthesis is a widespread property in prokaryotes. They are accumulated in the former intracellular granules when microbial cells are unable to grow at the same rate at which they uptake the available organic substrate. Growth limitation can be driven by external factors such as shortage of nitrogen, phosphorus, and oxygen. Indeed, microbiologists suggest that the main role of PHA is to store both carbon and energy, though PHA can also provide stress resistance against various environmental stress factors. While commercially, PHA are produced using pure cultures fed on sugar-rich biomasses, alternative low-cost processes are under development which make use of microbial mixed cultures, MMC, fed on second-generation organic sources, that are those organics that do not compete with the food or feed markets. Mixed microbial cultures capable of accumulating PHA are common in biological wastewater treatment processes, especially in enhanced biological phosphorus removal systems operated under dynamic feeding conditions. PHA can therefore be integrated in wastewater treatment plants by producing volatile fatty acids from the fermentation of primary sludge and using MMC from the biological section. When integrated in wastewater treatment plants, PHA production is typically operated in 3 separate stages. First, the acidogenic fermentation stage for VFA production, then the culture selection phase, and finally the PHA accumulation phase. Recent demonstration projects of the integration of PHA production in wastewater treatment plant include the SMART plant and the FARIO projects, which are providing operational data for a reliable assessment of production costs and environmental benefits.
Extracellular polymeric substances are sticky polymers secreted by bacteria and other microorganisms. Their production helps to bridge bacterial cells and other particles into aggregates. The extraction and recovery of EPS from granular biomass is at present the most interesting and developed opportunity, thanks to the ease of extraction and the high amount of EPS present, up and above 50% on a dry basis. Granular biomass is an established technology in anaerobic treatment of industrial wastewater and to remove nitrogen by Anamox process. The treatment of municipal wastewater with aerobic granules is more recent, but it's developing and spreading fast. Extraction of EPS from granular sludge can be achieved by a quite established combination of physical, chemical, or biological methods. Potential applications of EPS extracted from granular sludge are huge: biosorbents or bioflocculants, flame retardants, curing agents for cement production, protective agents in industrial applications, and more. The potential market supply for EPS extracted from biological granules could be quite relevant. As an example, it was estimated more than twice the national demand of alginates in Holland. A commercial product based on the EPS derived from a wastewater treatment technology has been already registered with the name of Kaumera Nereda gum. We studied the application of EPS extracted from anaerobic and Anamox granules in paper manufacturing and coating, and partial substitution of the traditional carboxymethyl cellulose. Waterproof and grease resistance of paper were measured in standardized tests. A very good increment, up and above 60%, was achieved for both properties when EPS were added. The recovery of polymers from wastewater is well underway. It will shape the future of wastewater resource recovery facilities, together with the other opportunities described in this massive open online course.